Hello, anatomy students. Happy Monday. Today we are going to just keep on trucking through chapter 26 notes. Last time we were together, we learned all of the different structures of the kidney. This is considered more of the gross anatomy or kind of the big picture of the kidney. And so let's go through each of these parts one more time. Remember that the nephron is the functional unit, and this is greatly exaggerated. This is probably 100 times larger than it would be in a real kidney. These are actually microscopic, and nephrons are what we're going to talk more specifically about in our notes today. A is going to be this outer functional layer. Remember, it looks like a letter C, so this is actually the cortex. B is going to be the medulla. So many organs we've talked about this year have the same structure of an outer cortex and an inner medulla. C is pointing to a little extension of the cortex that goes between D, our renal pyramids, and so we call this the renal column, which makes sense because it's like you think of a column on a building or something. And then we have our capsule is the outer layer, and don't forget that a single renal lobe includes a pyramid and the adjacent half of a renal column. So this particular kidney would have 1, 2, 3, 4, 5, 6 renal lobes. So that's kind of the gross anatomy of our kidney. Today we're going to talk more about the microscopic anatomy, or exactly how kidneys filter blood and make urine. Just a quick overview of our weekly schedule. Tonight is parent-teacher conferences in the gym from 3:30 to 9, so go ahead and send your parents or come with them. I will plan on being back for those if you need to come and visit. Wednesday is going to be our kidney dissection. So remember, whenever we do dissections, we need closed-toed shoes, a hair tie if you have long hair, and if you wear a sweatshirt, either be able to push those sleeves up or wear something underneath so you can take it off. Nothing we use is dangerous, it just stinks, so I don't want you to get any of that solution on your nice school clothes. Also, Friday is a day off, so we have that to look forward to as well. So the urine is formed in those nephrons, and we'll talk more about this specific structure of the nephron in a few slides. But as it is produced, it's going to drain into successively larger structures. And so as we go through each of these structures, we will go back to that original picture and label them, so hopefully they make a little more sense. So first is the renal papilla. A single papilla will drain each nephron into the minor calyx, and the minor calyx is kind of a cup at the point of each of those renal pyramids. At this point, once we get into the minor calyx, the urine is not changed at all. There's no more filtration or reabsorption, no more adding or removing materials. It's essentially just making its way out of the body at this point. Sometimes the minor calyces will join into a major calyx, which all then do converge into the renal pelvis, and then that will thin out and become the ureter. So remember, we do have two kidneys, and they're kind of mirror images of one another, and those renal pelvises are going to be near the hilum of the kidney, and those ureters then will drain on the medial aspect of each kidney. So here is that structure, starting with our nephron. So most of the filtration is going to happen at this little bulb at the beginning of the nephron, called the renal corpuscle. Then it's going to make its way through this winding tube, and then eventually whatever is left in the nephron will be excreted. Once it gets put in the renal papilla, which is just this kind of whitish region at the bottom where all those nephrons drain into the little cup here called the minor calyx. You can see if we have 1, 2, 3 minor calyces, and they all drain into this one spot called a major calyx. Then all the calyces will combine into this central region of the hilum called the renal pelvis, and it looks almost a little muscular, and it is. There is smooth muscle in all of these structures that will kind of squeeze or push that urine down toward the bladder. Excuse me. And so eventually it will narrow out and become the ureter. So the blood flow through the kidney is really essential because that's the main job of the kidney, is to regulate blood composition and blood pressure. So they can filter around 1 liter of blood per minute. So this guy right here is about half a liter. So 2 of these per kidney per minute. That's a lot of blood. that is being filtered. The blood is provided to each kidney through the renal artery, which is very large. It branches right off of the abdominal aorta. And then you can see how it branches into smaller and smaller vessels. So the renal artery will branch into segmental arteries. And you can think of that as going to different parts or different segments of the kidney itself. Then it will go into an interlobar artery. So interlobar, remember what we learned yesterday, that a lobe is just a single renal pyramid and its adjacent renal column. And so think of this as going between two lobes. So it's going to travel right up that renal column. Then it's going to arc around the top of that renal pyramid. It's going to kind of take a turn. So we call this the arcuate artery. And then finally, it's going to go into the renal cortex. So the cortical radiate artery is kind of the last step before we're finally getting to the nephron itself. So from there, we have the afferent arteriole. This is the same structure that we saw in our lymphatic vessels, or those lymph nodes rather. So remember that the afferent is going to bring the material into the structure. In this case, it's an arteriole, so it's bringing in blood, and the efferent will leave. In this case, all that's happening here really is the filtration of fluids. And so notice that it's still an arteriole both times. The actual gas exchange and material exchange that keeps the kidney alive is going to happen via the vasa recta and the peritubular capillary. So these are both arterioles, and what's happening in that glomerulus, there we go, the glomerular capillaries, is kind of the drawing out of excess water and waste material. The actual kidney itself is kept alive by the vasa recta and those peritubular capillaries. So these words seem kind of complicated, but the good news is when we're talking about the drainage, after all of this filtration and the dropping off of nutrients and energy have happened for the kidney tissue itself, the draining is exactly the same, except backwards. And so we start with our cortical radiate veins through, there we go. So it's going to drain material, and then it's going to go around the corner of our renal pyramid. So we call it the arcuate vein. Then we have our interlobar vein, and then we have the renal vein. So for some reason, the one they skip is segmental. I'm not exactly sure why, but other than that, this structure of drainage is exactly the same as the order that we, bring materials into the kidney. So like we mentioned, fluid and solute exchange is happening, and additional exchange to keep the kidney tissues alive and oxygenated are going to happen in these peritubular capillaries. So blood flow to the kidneys is really abundant. It's where a lot of your blood is at any given time, and it's going to be regulated by your sympathetic or your autonomic nervous system. When you're in fight or flight mode, this is actually one of the main ways that we can increase blood pressure is by constricting the renal arteries, and that allows for all of that blood to be used elsewhere, like in your nervous system, in your skeletal muscles, or to keep the heart and lungs pumping and getting enough oxygen to the rest of the body. So remember that sympathetic is your fight or flight division. Then when you come back down that fight or flight reaction in your parasympathetic mode, then we're going to dilate these renal arteries so that we can kind of catch up, make up for lost time, and get rid of any excess waste that was produced during that fight or flight response. So these reactions are just autonomic. It's nothing that we can consciously control, but a really important part of regulating how quickly the kidneys do their job in creating urine and filtering your blood. So now we're going to zoom in even closer to the components of an individual nephron. We mentioned those little bulbs where the blood flow kind of comes into each nephron is called the renal corpuscle, and it contains a capillary network called the glomerulus, and then a little connective tissue capsule called the glomerular or Bowman's capsule. This little cup is going to draw fluid in and then enter the renal tubule. This tubule has a couple of different sections, and each section has a different amount of porosity. So some parts will draw more fluid in from the kidneys tissues, and some are really good at allowing more material to be reabsorbed. And they're all sensitive to different types of materials. So let's just take a look at these, because I think the diagram makes way more sense than our definitions. And so again, this is our afferent arteriole. It's going to come in, and this is where the blood will stay. Notice that it's only fluid that's passing from the glomerular capillaries into the capsule. Then it's going to twist and turn through the proximal convoluted tubule. So we know what a tube is. Proximal means it's closer to the beginning, which makes sense. And then convoluted just means it's kind of twisty and turny. So after the proximal convoluted tubule, we have this nephron loop, which goes down, so it's descending, and then it comes back up, it's ascending. We sometimes call this the loop of Henle. Then we have the distal convoluted tubule, which again, it's twisty and turny, but this one is farther away from the glomerulus. So hopefully these names make sense to you. It's a little throwback to those anatomical terms. The last step in our nephron is the collecting duct, which will then drain into that renal papilla we started the day talking about. So when we look at the length of that nephron loop, there really are two types of nephrons. The most common type are cortical nephrons, which are just like the one on the previous slide. These produce dilute urine all the time, and they really don't go down very deep into the kidney. But in between 5 and 20% of all the nephrons, depending on your genetics and whether you're male or female, are called juxtamedullary nephrons. So we had this word juxtamedullary last week, and it means that it is close to the medulla. And so these nephron loops are really, really deep. And the deeper we go into our kidney tissue, the higher the gradient that is produced between the tube and the kidney tissue. And so this can produce really dilute urine if you are very hydrated and you have lots of water to excrete, or it can produce very concentrated urine if you are very dehydrated, but you still need to get rid of this, you need to get rid of the metabolic waste. So here you can see them side by side. The cortical nephron has its glomerular capsule up higher in the cortex, or less deep, more superficial. And then our juxtamedullary nephron has its corpuscle deeper in the cortex, and its loop dips all the way down to those deepest and saltiest layers of the medulla. So juxtamedullary because the corpuscle is right next to the medulla. The border between the medulla and the cortex is this dotted line here. So what's happening in the glomerulus or throughout the nephron are three processes. Filtration is going to happen in the glomerulus, and this is the initial moving in of all the materials that are water soluble into the nephron loop, and now it is urine. Then throughout the tube, we will reabsorb some materials, but also secrete some materials. And so through these three processes, we then create the material that is excreted via your urine. So we want to keep things that are good, things like salt, things like proteins, glucose. These types of materials will be reabsorbed in the nephron loop. We want to secrete things like urea or any metabolized medicines or drugs. And so this initial glomerular filtration essentially takes everything out. I think in the crash course, what they say is it's kind of like cleaning out your fridge, where you take everything out and then you only put back in the things that you want to keep. And then anything that's not put back in, or reabsorbed in this case, is excreted. So it looks like we're just about out of time here, so I'm going to skip ahead a few slides. So just a reminder of what you are to do today. You can rewatch and redo this quiz as many times as it takes to get a good score. It will end up in the gradebook for quarter 4, believe it or not. Quarter 4 starts today. If you have time left over, you can work on your histology or your chapter 26 homework. Make sure you're drawing, coloring, and labeling. And I will be back tomorrow. So we will connect then and continue talking about kidneys.